So now we want to move on to the extracellular space and immune response against extracellular bacteria. Okay, I stand corrected. Not against extracellular bacteria, against extracellular pathogens. Again, I correct myself, not against extracellular pathogens, against pathogens when they are located in the extracellular space. That is the correct thing. Exactly. Because when I say extracellular pathogen, what it means is that pathogens which live in the extracellular space. But that is not entirely correct. Immune responses in here, which are mostly antibody responses, can influence pathogens which live in the extracellular space, as well as the pathogens whose final destination is intracellular. But during the process of infection, they have to traverse, they have to go through the extracellular space. And this is very, very important. Because if we do not consider the path, PATH, the path of the pathogen, then we say, okay, for intracellular pathogens, they are located inside cells. Antibodies do not have access to the intracellular space. So humoral immunity would be irrelevant. Humoral immunity is never irrelevant because all of the pathogens have to go through the extracellular space. Sure, exactly, exactly. Even that's a very good comment. Even if it's a virus which is going to become intracytosolic, after its release from the target cell, it again enters the extracellular space, so it can get re-exposed to humoral immunity. That's very good. All right. So this is an extremely important area of defense, and the majority of the vaccines that we have, vaccines against different bacteria and viruses, with one major exception, which is BCG vaccine. They protect us by induction of antibodies, by induction of the production of antibodies. Of course, T cells would have to support those B lymphocytes in the production of antibodies, as I will show you later. But antibodies would be the effector molecules. All right, so this is a slide that you have seen something, this slide or something similar to this before, just to refresh your memory about B cell activation. We have B cells produce antibodies. These antibodies can perform their function in different ways. They can neutralize pathogens at the surface of mucosa if they can be secreted, exactly. They can neutralize pathogens like viruses or bacterial toxins in the extracellular space, in blood plasma, for instance. We have another process. If you cannot neutralize it, you can facilitate the phagocytosis. It's called opsonization. You can activate complement, a cascade of proteolytic enzymes, exactly, which is present in serum, and it can perform different functions. You can activate the so-called antibody-dependent cytotoxicity, bind to the surface of the cells, and then NK cells and neutrophils can perform that function. And you can also activate eosinophils and mast cells and induce their degranulation. And we have different classes of antibodies for them. IgA performs mucosal neutralization. IgE is the one involved in degranulation of eosinophils and mast cells, and then the rest of the story. So one thing to keep in mind is that here we have IgM, which is the first antibody which is produced by a B cell when it gets activated. It's a very good activator of complement. It can also neutralize things, but it doesn't perform all of the functions of the subsequent antibodies like IgG. IgG is a very good activator of complement. It opsonizes very well, and it's a very good neutralizer. So if I'm a B cell and I'm producing IgM, well, I'm doing my function, but it's better for me in the majority of scenarios to produce IgG. So this is what we're going to see now. If you have a B cell and that B cell is alone, no help from any other cell, and it sees the antigen, it can respond to the antigen, but it usually produces IgM. IgM is a very good antibody, but it doesn't perform all of the functions that we require from antibodies. So we need further help. And one of the processes that helps B cells is the T cell that has been already activated by an antigen presenting cell. I mentioned that when something is phagocytosed by an antigen presenting cell, like a macrophage, or endocytosed by a dendritic cell, regardless of whether it's a hard to kill or an easy to kill pathogen, the antigens are presented. Well, we have co-stimulatory molecules as well. I haven't drawn them in here. And at certain circumstances, interleukin 12 is also produced. T helper 1s can produce interferon gamma, which could help the antigen presenting cell, especially if it was a macrophage, to to produce reactive oxygen species and eliminate things which are like mycobacteria. They're hard to kill entities. But in this case, I'm not dealing with a mycobacterium. I'm dealing with a pathogen which lives in the extracellular space, but it has been phagocytosed by macrophages. The T cells get activated. We have T helper 1 cells, and then they produce interferon gamma. So the interferon gamma in here is of no use for the antigen presenting cell, but it can influence B cell and induce the production of IgG. Exactly. This is the process which is called class switching of antibodies or isotype switching. It's one of the major aspects of B cell biology. Why is this process important? Because if you don't activate it, the only thing that B cells can produce is IgM, and IgM does not have a lot of properties that we require. So this is extremely important process. And in humans, we have 4 subtypes of IgG. We have IgG 1, 2, 3, and 4. For after interferon gamma production, it's mostly IgG1 and 3 which are produced. IgG2 is produced at a lower extent, and IgG4 has a different story that I will tell you later. So this is one process. This is important when we require IgG. I will show you about the situations, I will show the situations that we require IgG. Another situation is when you are dealing with an entirely different class of pathogens, especially helminths, worms, and some other eukaryotic parasites. So if you're dealing with a worm, a helminth, or in the majority of cases, some other eukaryotic parasites, production of IgG would not be helpful. Production of IgM would not be helpful either. You have to help T cells differentiate to another subtype. And how do you do that if you present antigen to a T cell, but you deprive it from interleukin 12? If you don't give it interleukin 12, by default, it starts going through differentiation process, which leads to the production of T helper 2 cells. T helper 2 cells can produce interleukin 4, which accentuates the process. It induces the production of T helper 2s in an autocrine as well as a paracrine manner. So you present the antigen to T cells, you don't give them interleukin 12, they start going towards T helper 2 very slowly. When the first T helper 2s are generated, they start producing IL-4, and that IL-4 accentuates the process, increases, augments the process, and then a lot of T helper 2s will be produced. So their interleukin 12 will be able to influence B cells, helping them to switch the class of the antibody to IgE, because this is the type of antibody which we require for defense against those parasites. We have a third scenario as well, and that third scenario usually happens when the antigen presenting cell is located beneath the mucosa. It's a submucosal antigen presenting cell. So it phagocytoses something, but it understands that whatever I phagocytosed, it has come from a microbe which is located inside the lumen of the intestine. If it's a if it's a mucosal, if it's an intestinal mucosa. So it needs to tell the B cells to produce another type of antibody. But here you see there is a hierarchy. It's like armies that you have a central commander at top, and then you have the top officers, and then you have lower ranking officers, and then you have soldiers. So the central commander directly talks to the high ranking officers. Who are the high ranking officers? CD4 T cells are the high ranking officers. So it tells them what to do, and those high ranking officers will communicate with the lower ranking officer in my analogy, which is B cell in here. So it produces some sort of cytokines, a mixture of cytokines, which induces, which promotes the differentiation of T cells toward another phenotype. Some people have called it T helper 3. Some people have said, no, don't call it T helper 3, just call it a subtype of CD4 T cells. Regardless of what we're going to name this type of T cell, they can produce TGF-beta. And TGF-beta can stimulate the production of IgA. Okay, so this is a bit lower than where it should be located. All right. So it stimulates the production of IgA. And IgA is what can be basically secreted. It can traverse the epithelium and it goes inside the intestinal lumen, the GI lumen, or it can also be produced in the respiratory tract to lower levels and can be secreted. So we have these three different defense mechanisms. So the first one is very useful when you're dealing with something in the serum or plasma or extracellular space, which requires neutralization. So, like bacterial toxins or viruses can be neutralized, or it requires opsonization, facilitate phagocytosis, or it requires complement activation. How about IgA? Well, it's very important when you need mucosal defense. How about IgE? It's important when you're dealing with something which cannot be neutralized or opsonized, or complement doesn't work against it either. So you have to cover it with IgE. These are IgE. You have to cover it with IgE. And then you have cells which have receptors for IgE, including eosinophils and mast cells. So when they see a lot of IgE, they go and bind, and they are ready to release their granules. However, again, they require a second stimulus, or a second factor needs to be involved in the process, just to be sure that they are not releasing their granules when it's not necessary. Okay. In general, when immune system is using a very deadly weapon, it checks twice. If it's an NK cell and it wants to target a potentially virus infected cell, it needs two checks: lack of MHC molecules and presence of stress antigens. Exactly. So in here, we have a similar situation. You need to have IgE molecules as well as some help from interleukin 5, which is, again, is produced by T helper 2 cells. So they release their toxic granules. Their granules are helminticide. Helminticide. Okay, so we have different terms in here. Helminticide. So they have the ability to kill worms, basically, and they can eliminate the worm. Mast cells also have some granules which contain things like histamine and some other peptides. So they are involved in the process, but their effect is slightly different. And here, well, we need a bit more specification. So we have two groups of worms. Some of them live at the surface of the intestinal epithelium, for example. Uh, they do not penetrate the tissues. They are not they don't live inside tissues. So there you have the involvement of mast cells is kind of more important to induce the production of mucosa and to induce the intestinal motility so that the worm is kind of removed. But if it's a worm or a parasite which is inside tissues, it's inside tissues, then the activity of eosinophils would be more important. So here we have defense against helminths. And you see one molecule in here, and I want you guys to go and study about it. We have one subtype of IgG, which is under the influence of interleukin 4. And this is a different type of IgG. And I want you guys to go and read about it. By the way, interleukin 5, I haven't shown it in here. Interleukin 5 also plays a role in the production of IgA. So interleukin 5 is an eosinophil activator, but it can also help B cells in the production of IgA together with TGF beta. I haven't drawn it in here. All right. So these are three different pathways of T cell activation and B cell stimulation. It was, I think, in 2009 that another group of T cells was discovered, and it was named T helper 17. It was T helper 17, and that's a sort of a peculiar naming. Okay, so we had 1, 2, and maybe 3, and then you all of a sudden you jump to 17. How about 4 and 5 and 6? So the reason that it was called T helper 17 was that before the discovery of T helper 17 molecules, a cytokine was discovered, and that cytokine, the naming of the cytokines 1, 2, 3, 4, it was named IL-17. And that IL-17, people recognize that this is a very strong neutrophil activator. And it's a neutrophil activator. But nobody knew what is the cell that produces it. It was named an orphan cytokine. It was named an orphan cytokine. So we use orphan in different scenarios, like when you say orphan drug, it's a drug that doesn't have a very good market, so nobody produces that drug. It's called an orphan drug. The number of patients which require it are so low, so pharmaceutical companies are not interested to produce it. It's called an orphan drug. When you say an orphan cytokine, it's a cytokine that you don't know which cell is producing it. And when T helper 17s were discovered, people discovered that you need a mix of cytokines. You need a mix of interleukin 1, 21, and 23, and IL-6 and TGF beta to produce these. They recognized that these are the cells which are producing IL-17. For that reason, they were named Th17. Like in Arabic, you have sometimes that the father is named after the son. Okay, so you say, like Abu Hassan, it means someone whose son has the name Hassan. This is like Abu IL-17 in this case. So it's called T helper 17 because it produces IL-17. So T helper 17s produce IL-17, they activate neutrophils, but they also produce another very important cytokine, interleukin 22, which stimulates epithelial cells. Okay, so epithelial cells are not, except the physical chemical barrier that they provide, they are not considered as leukocytes. They are not leukocytes. They're epithelial cells. They produce the surface lining of different tissues. Nonetheless, they have a very important ability, and that ability is that they can produce antimicrobial peptides. AMPs, antimicrobial peptides, but they don't produce it under normal circumstances. So you need to stimulate them with something to tell them that you guys need to basically start producing. You need to take up your weapon and defend the boundaries of the body against the pathogens. And that stimulus comes from TLR17s. It's interleukin 22. So this has two sets of roles in here, stimulating neutrophils and epithelial cells. So this is the structure that you're seeing in here. So now I want to use a metaphor. Metaphors always help. And that's the metaphor of a classical army. It's the metaphor of a classical army. So a classical army usually has a central commander. The central commander is usually a very old soldier, a veteran soldier that has fought in different battles, is very good in fighting, but is also very good in understanding and devising military strategies. So that's the central commander. It tells other people what to do. Then you have high-ranking officers, which are beneath the central commander. They are also very good soldiers. They're very experienced, but they are not as good as the central commander in devising military strategies. Then you have low-ranking officers. Then you have, okay, so in a classical army, the highest number of personnel are regular soldiers, ordinary soldiers. So you have one central commander, maybe you have 100 high-ranking officers, you have a couple of hundred low-ranking officers, but then you have thousands of soldiers. You have thousands of soldiers. And then you also have people, civilians, ordinary people, especially people who are living at the borders, because that's the region that gets attacked first. So if you want to compare this structure with a classical army, who is the central commander in here? The antigen presenting cell is the central commander. It's a very good soldier. It can fight very well, performs phagocytosis, but it knows how to activate different immune defense mechanisms. Do we need to activate the T helper 1 pathway? Do we need to activate the T helper 2 pathway? Do we need to tell the cells to produce IgA? Do we need to tell them to become T helper 17? That is what is recognized by the antigen presenting cell. It recognizes the class of the pathogen. How does it do that? It has a lot of receptors for it. Exactly, PAMP recognition receptors or pathogen recognition receptors like TLRs and NLRs and CLRs. So it has a set of molecules which are evolutionary old they have evolved during millions, hundreds of millions of years. And that those receptors enable the central commander, which is antigen presenting cell in here, to understand the situation. And then based on that situation, it can activate different types of cells. Who are the high ranking officers? Who are the high ranking officers? CD4 T cells. CD4 T cells are the high ranking officers. These are T helper cells, and they sometimes become T helper 1 or 2 or maybe 3 or 17. So these are the high ranking officers. Who are the low ranking officers beneath them? B cells and CD8s. We can, in this analogy, in this particular type of analogy, we can call them lower ranking officers. So they listen to CD4 T cells. Sort of, sure, you can say it that way too. Yeah. So they tell the B cells, okay, you need to produce IgA or IgG or IgE, and they also activate CD8 T cells. I didn't show you the process of antigen cross presentation, but they are involved in the activation of CD8 T cells as well. Finally, we have the soldiers, the regular soldiers. These are ordinary soldiers. They fight very well. Their number is very high. If you let them fight by themselves, they are okay. But if you have an officer which guides them and activates them and stimulates them and motivates them, they work better. Who is that officer? T helper 17s. So T helper 17s deal with regular soldiers as well as the civilians. These are the civilians. These are non-military people. Which kind of civilians? The ones that live at the borders, because that is the most sensitive area. So it tells them that, okay, you guys can also take up arms and start defending, because it will take time for the regular soldiers to come and the rest of the things to get activated. So this is the process. And then we have these mechanisms in here, interstitial space. We have T helper 1, interferon gamma, B cells, and IgG. Here, if it's a parasite, we have T helper 2, IL-4, B cell, and IgE. Here at the epithelial cell place, we have another type of T cell. Some people have called it T helper 3, TGF beta, B cell, and IgA. Here again, we have T helper 17s, IL-17, and neutrophils. And here we have T helper 17, IL-22, epithelial cells, and antimicrobial peptides. So these are different levels of defense. And as you see, this is a very highly structured system. And it should be very difficult for a pathogen to get through these things. So before I continue, I want you to write down what I said. Okay, because we're moving to evasion mechanisms, I'm going to save this session, and then we'll start from evasion mechanisms.